mediated tumor cell microbiology model. The pH level in solid tumors is reported to be more acidic than normal tissue, with values 6.2 through 6.9 than in normal tissue 7.2 through 7.4, which is attributed to enhanced glycolysis and lactic acid secretion, a condition referred to as the Warburg effect. The acid pH level surrounding tumors has been linked with increased invasion metastasis as well as lower treatment response. The catalase is another antioxidant enzyme involved in the conversion of hydrogen peroxide to water and oxygen. The enzyme is pH dependent and has maximum activity at pH levels near neutrality and slightly basic. The objective of this research is to evaluate a tumor-like and normal tissue pH level using a safe potato catalase model to understand how pH levels might affect catalase-based anti-cancer therapy for tumors. The research question is, how does tumor-like acidity affect the ability of catalase to break down hydrogen peroxide? Purpose. The purpose of this study is to determine how tumor-like acidic pH conditions affect the ability of catalase to break down hydrogen peroxide compared to normal physiological pH in order to model how the acidic tumor microenvironment may influence catalase activity. Our hypothesis is the catalase activity will decrease under tumor-like acidic pH conditions compared to normal physiological pH, resulting in a slower rate of hydrogen peroxide breakdown as acidity increases. Potato tissue will be used as a source of catalase, with uniform potato discs prepared to ensure consistency. The discs will be placed into hydrogen peroxide solutions of equal concentration that have been adjusted to different pH levels representing tumor-like acidic conditions and normal physiological pH. As catalase breaks down hydrogen peroxide, oxygen gas will form and cause the disc to rise to the surface. The time required for each disc to float will be recorded as an indirect measure of catalase activity, with shorter times indicating higher enzymatic activity. Multiple trials will be conducted at each pH level while controlling variables such as temperature, disc size, and hydrogen peroxide concentration.
Data table, which can be seen on the left side of this slide. Our data showed that under tumor-like acidic conditions, catalase activity decreased as pH became more acidic. At pH 6.2, the potato discs took the longest to float, with a mean of 85 seconds, indicating the lowest rate of hydrogen peroxide breakdown. At pH 6.6, the discs floated faster, but were still slower than normal conditions, with a mean of 60 seconds. The fastest reaction occurred at normal physiological pH 7.4 with a mean of 32 seconds, while slightly basic pH of 8 showed similar or slightly reduced activity with a mean of 35 seconds. With this, our results are that overall the results show an inverse relationship between acidity and catalase activity, supporting the hypothesis that tumor-like acidic pH reduces the enzyme's ability to break down hydrogen peroxide. Next, for our reflection. This project helped us better understand how enzyme activity is affected by environmental conditions and how this concept connects to real-world cancer biology. By modeling tumor-like acidic pH using a potato catalase experiment, we learned how changes in pH can significantly influence reaction rates and enzyme function. The lab also strengthened our experimental skills, including controlling variables, collecting reliable data, and analyzing results through graphs and averages. We gained a greater appreciation for the challenges of modeling complex biological systems, such as tumors, with simple laboratory tools. Overall, this project showed us how basic biochemical principles can be used to explore important medical questions and helped us develop critical thinking, teamwork, and problem-solving skills. Next, for our future improvements. For future improvements, this project could be expanded to better reflect real tumor conditions and strengthen the reliability of the results. Using a pH meter instead of pH strips would allow for more precise and consistent pH measurements, reducing experimental error. Increasing the number of trials and replicates at each pH level would also improve the statistical reliability of the data. Additionally, testing catalase from different biological sources, such as animal tissue, could help compare how enzymes from various organisms respond to acidic environments. Future studies could also incorporate additional variables like hydrogen peroxide concentration or temperature to better model the complexity of the tumor environment. Overall, these improvements would help make the experiment more accurate and deepen the connection between enzyme activity, pH, and cancer biology. And lastly, for our acknowledgments, we would like to acknowledge and give our best regards to our PLTW Biomedical Innovation Mentor, who allowed us to utilize his lab materials as well as provide outstanding support throughout our Capstone project process. Additionally, we would like to acknowledge our chemistry mentor, who worked concurrently with us to establish a strong foundation of understanding pH levels as well as dilutions in order to develop our base and acid solutions. And that is it for our project. Thank you so much for listening.